Excuse me, bro.
Yes?
I can't record. You didn't give me the permission.
Lecture notes. These are lecture notes.
Okay, there you are.
In front of you.
Okay. You have the right now? Okay.
Yes.
All right. And Axilla, you have a lot of background noise. I hope it won't be recorded in the recording. So just tell the people in the background to tone it down. So let me see. I want to enlarge this screen. So for those people who came in late, my lecture will end by 10 so that I can join another meeting that I was not excused from. I really wanted to finish up today, so I will give you the recordings for what I wouldn't have covered. So for now, I wanted us to look at synaptic transmission, the initial stages. If you look at from the time you initiate, why isn't it showing? Let me see again. There you are. So if you look at the steps that are happening before the synapse is that gap that connects the neuron to the adjacent neuron. So for the synapse, you have two neurons that are communicating to one another. So in the presynaptic neuron, all the events are similar in the neuromuscular junction. So there is an action potential that is coming, depolarization of that presynaptic membrane, which allows the calcium voltage sensitive channels to open and allow calcium to flow in. The purpose of the calcium is to bind to the presynaptic vesicles so that they can also dock to the presynaptic membrane and initiate exocytosis, releasing their contents into the synaptic cleft. So all those events are the same even in the neuromuscular transmission. Kindly note, and this is very important, the neurotransmitter for a neuromuscular junction is always acetylcholine. The neurotransmitter, because you're talking of the release of a neurotransmitter. In a synapse, it could be anything, and we'll give a list of all the neurotransmitters that you can release in a synaptic cleft. However, in a neuromuscular junction, the neurotransmitter is always acetylcholine. When acetylcholine binds to the postsynaptic receptors, it usually causes opening of sodium channels, and because of that, it will cause depolarization on the postsynaptic side, and that depolarization can trigger in a postsynaptic neuron, it can trigger an action potential as shown here. Note one difference again, in a neuromuscular junction, we don't have a postsynaptic membrane. We'll look at what we have instead. Now, the receptors there are also channel receptors, and they allow sodium to flow through them. So there's voltage sensitive sodium channels. So upon binding, now in the postsynaptic region or post-neuromuscular region, on one side is a neuron and the other one is a muscle fiber. So neuromuscular junction talks of communication between a neuron and a muscle. So in that case, because it is acetylcholine and it will allow sodium to flow in, the resultant effect on the muscle is different from a postsynaptic neuron, because the end result will be a muscle contraction. For this one, the end result will be an action potential that is initiated at the axon hillock. So that is one difference. So it is good for you after I go through the neuromuscular junction events to look at the difference between a synapse and a neuromuscular junction to know the major differences, and if you are asked, you can enlist them. So this is similar to the last diagram I had shown you. We did this in class, showing the synaptic transmission, the events that happen. Now, as we talk about synapses, what are they? These are junctions between two neurons. One is the presynaptic neuron and the other one is the postsynaptic neuron. And there is typically a gap between those two, and that gap is what we call the synapse. Now, it is also important as we discuss the synapses that we note there are two types of synapses. You can have the electrical synapse or you can have a chemical synapse. The events that we have been describing involve the release of a neurotransmitter, and the neurotransmitter is a biomolecule, so it is a chemical. So in that case, we were discussing the chemical synapse. For the electrical synapse, we do not have such a gap. These are actually enabled by what we call gap junctions, and I'll come to that when we are looking at signal transduction. You end up having two adjacent cells. They could be neurons or they could be otherwise. And what happens if these are two cells? You can have something like a tunnel. You see, you can have a tunnel here which will allow continuous flow of cytoplasm from one cell to the other. Now, this is what we call a gap junction. So, actually, what will happen is when you depolarize this cell, you find that you are going to initiate some of these gap junctions to open their channels. Remember when we were discussing about how you can transport things across the cell membrane? So there are membrane channels which are connecting the cytoplasm of cell A to cell B. So when you open those channels, then whatever ion or electrolyte they are getting or they are allowing to pass will pass directly from one cytoplasm to the other. For that reason, it's a charge that is moving across the cell membranes. And since charges are the basis of the electrical events, we say that is an electrical synapse. So this differs from the one we have just discussed, which is the chemical synapse. For the chemical synapse, we say there is a biomolecule which is released in the cleft, and that biomolecule is a neurotransmitter, and it is the one that carries the signal across a chemical synapse. Now, for this to happen, we must have an interaction between the neurotransmitter itself and the receptor on the postsynaptic membrane. And that combination is the one that will trigger events in the postsynaptic neuron. Those events could either be depolarization, meaning they are stimulating the second cell, or it could be hyperpolarization, in which case you say they are inhibiting the second cell. So the neurotransmitter receptor combination determines the events that happen in the postsynaptic neuron. Now, the interesting thing is that a neuron will always release the same neurotransmitter. It won't change. So if it's releasing acetylcholine, every time you stimulate it, it will always release acetylcholine. If it is releasing a different neurotransmitter, say epinephrine, then it will always be releasing epinephrine. Now, when we have a presynaptic neuron releasing the neurotransmitter, you find on the postsynaptic membrane, we have a receptor for that particular neurotransmitter. For that reason, every time you stimulate that neuron, the events will always be the same because they will initiate that combination and the results will always produce the same response. Now, the only difference here is depending on how much neurotransmitter you release. if you release a little, say you only had one stimulation, then you release just a few vesicles, then it means on the postsynaptic neuron, the depolarization will be very slight. Or if in case we are talking of hyperpolarization, it will be minimal. If you increase the signal, the action potential signal in the presynaptic neuron, if you increase the signal, and do you remember how you increase the signal? How do you increase the signal in a neuron? You increase what, Lillian?
The frequency.
The frequency, thank you. So when you increase the frequency, it means that there is one action potential that hits a given number of vesicles, but before even those vesicles have cleared, another one has come in. It has stimulated more vesicles to go to the presynaptic membrane, and before those ones have even reached, another one has come. So what will happen is that in the synaptic cleft, we are going to release a massive amount of neurotransmitter. For that reason, the postsynaptic receptors will be bombarded with a lot of neurotransmitter. Therefore, in the postsynaptic neuron, you'll cause a massive depolarization, which is likely to trigger an action potential. Now, the other thing you must note in the synapse or neuromuscular junction, it is crucial, it is very essential that you remove those neurotransmitters from the cleft. If you don't remove them, they'll continue to stimulate that cell. So you want to remove them so that you can end the stimulation. So I will skip this, and you find that some of these things I may be browsing through them, because when we go to Physiology 1332, this is one of the topics where we have to compare the neurotransmitters and what they do. So although I have listed them here, the purpose of this table was to show you that we don't only have one type of neurotransmitter. In this case, we talked of acetylcholine. And do you remember which neurons always secrete acetylcholine? Who can remind me? It was the first thing I discussed today. Which neurons will always use acetylcholine as their neurotransmitter?
Is it then the motor neurons which terminate the muscles?
The
Yes.
neuromuscular.
Yes, thank you. At least you're paying attention. The neurons that innervate the muscles. So at the neuromuscular junction, you will always have acetylcholine. Now you can also have other neurotransmitters. I have mentioned epinephrine, and when you think of epinephrine, always think of norepinephrine. We also have others that are listed here. Some of them will cause stimulation, such as the ones I have mentioned, but some of them cause inhibition, such as this one. You have the GABA. GABA is an inhibitory neurotransmitter, and so is glycine. So these ones are inhibitory, they cause hyperpolarization. But we'll discuss this in greater details when we are doing Physiology 1332. So what I wanted to introduce today is the neuromuscular junction, to show you here is a neuron, just like in the synapse. It is bringing in an impulse, an action potential. And the events in the presynaptic area is exactly the same as we have described. Now, the difference that I said on the neuromuscular junction, at the postsynaptic membrane, it is not a neuron we have. This is a muscle cell. It's a muscle fiber. And this muscle fiber usually has a cell membrane. Now at the junction, that region of the junction, this cell membrane of the muscle fiber becomes modified. And when it becomes modified, it forms what we call a motor end plate. I don't see it recorded here, so I will just type for you, and I'm sure I'll see it somewhere also. What you'll end up having is a motor end plate. So this is a modified region of the muscular plasma membrane, And we say it is modified because it congregates, it brings in a lot of acetylcholine receptors. You can see these receptors here. They are actually, they congregate within that areas. And secondly, these acetylcholine receptors, they also double up as sodium ligand sensitive channels. So the ligand being the neurotransmitter. So when the ligand binds, it opens the channels, and those channels will initiate a depolarization here, similar to what we see in the neuron, in the postsynaptic neuron. So the difference is when we have this depolarization of the muscle cell, this will be transmitted interiorly into the muscle fiber. So the membrane potential is not just flowing on the surface of the muscle fiber. It also penetrates deep into the cell, into the muscle cell. And what happens is that it will initiate the events of muscular contraction. That will be a topic again in Physiology 1332. So what I will tell you, if you are asked to describe the events, you end with the depolarization of this membrane, causing depolarization inside the muscle fiber, resulting in a muscle contraction. So don't go to the details of what happens during contraction. Just end that the end point here is a muscular contraction. So with that, let me ask if anybody has a question on the neuromuscular junction. I told you I'll be very brief on it. Any question? If not, I will end this one. I will end this and I start on the next topic. Yes, Asna.
Excuse me, madam, when you're talking about the neuromuscular transmission, do you also include the part for the enzyme to break down acetylcholine?
Definitely. You always have to end. You end the events in the postsynaptic cell, what has happened to it. And number two, you also say what happens to the neurotransmitter. If you don't remove the neurotransmitter from the neuromuscular junction, that muscle will continuously be stimulated, and you don't want that to happen. Yes, Anyim?
Okay.
Or you have another question, Asna?
Oh, thank you.
Alice?
When you're told to describe the events that occur in a synaptic transmission, do you also say like there's a possibility it can come from an inhibitory synapse, or we just use the excited synapse?
Yeah, when it is a synapse, you always talk in terms of there's a possibility at the postsynaptic neuron, if the neurotransmitter is inhibitory, that it will cause hyperpolarization. Then you can mention if it is stimulatory, it will cause depolarization. You don't have to say which channels, because in some cases, the depolarization may be due to calcium channels. The hyperpolarization may be due to opening of either potassium channels or chloride channels. So you can mention them, they'll give you a bonus because it shows you have the knowledge. But if you just mention that in case of hyperpolarization, it will cause inhibition. In case of depolarization, it will cause stimulation in the postsynaptic cell. Okay, so today's topic was supposed to be how cells talk to one another. And I have put this slide showing you that I got this information mainly from human physiology, cells to systems. And I think I've given you this in your Blackboard. So you can actually go to this book. If it is not in your Blackboard, then you can get it from the library. I know it is there. So you can go through the system, I showed you how to retrieve the books, go for cell-to-cell communication. It's chapter 4 of this particular book, Cells to Systems. It's available, it's an ebook. Okay, so what does it tell you? One of the things that we discussed at the beginning of these lectures is that we have several types of organisms. Some of them are unicellular, others are multicellular. And our cells happen to be within the multicellular organism. And these organisms, the cells have to talk to one another so that they can maintain the internal environment, because the external environment is always fluctuating. It's always changing. So what the body has to do is to adapt to those changing environments. So how does it do? The cells have to talk to one another and let each other know what is happening outside there and how to adjust to each other. So we have to have this integrated system whereby different systems which play different roles talk to one another so that they achieve the same function, which is to maintain the internal environment. So how do they talk to one another? There are two ways. Cells can talk between themselves. So we have cell A talking to cell B. And we have different ways in which that can be achieved. Now, I want you to note that when cells, different cells are talking to one another, then in that case you talk of intercellular. Inter means between, signal transduction or signal communication. This can occur in different ways. Just give me a minute, please. Okay, sorry about that. So as I was mentioning before, is when cells talk to one another, there are different ways in which they can talk. There are these signaling, we talk of autocrine, paracrine, endocrine, all these. I'll go to the details later. What I want you to note is that inter means between cells. Although in the first one, you can see it is the same cell talking to itself. The second type of communication, when it comes to giving information to the cells, it's the events that are happening outside a particular cell. And those events are interpreted inside the cell. So information is coming from outside the cell and events or responses are inside that cell. So this is a second type of transduction. You call it intra because it is inside the cell, intracellular signaling. So we shall start with the intercellular and then we'll finish up with the different methods of the intracellular signal transduction. So you can have, during the intercellular, between cells, you can have cells that talk to one another by direct communication. Cell A talking to cell B directly, and I have mentioned one of them. Who can remind me what type of signaling or the type of communication that I had just talked about, where two cells talk directly to each other?
Intracellular.
Yes, but I remember I gave an example.
Using the gap junction?
Gap junction, yes. Oh, even it's written here. You can have direct intercellular communication between cells through gap junctions. And this is because it has a tunnel that is connecting one cell to the other cell. or you call them, in this case, they are talking of nanotubes, very tiny, tiny tubes, which are actually the channels that we are talking of. Alternatively, if it is not through the gap junction, it could be through surface markers. Each cell is unique. It has a given number of markers. You may have proteins, glycoproteins, lipids, and all that, and they are all protruding from the surface of the cell. These are very unique. Now, when it comes to a different cell, the different cell may have so we have the different cell which is adjacent to it, and it may have receptors that can actually bind these markers. And when they bind the markers, they cause events to happen in the adjacent cells. So this is by link up of the surface markers. Then this one will talk of what kind of communication it is. So it can either could either be through the gap junctions. So I can draw a gap junction here. So you can have gap junctions which allow direct communication. So, or you can have the surface markers which allow direct contact and therefore initiating. So this is the signaling cell, and this is the receptor in case of the gap junction, and this is the receptor in case of the direct communication. Now, it doesn't mean that if this is the signaling receptor, the signaling cell, you cannot have it being a receptor. It can also be a receptor because this may have surface markers, and it has this one has a receptor for it. So in such a case, this will be the signaling molecule, and this will be the receptor. Why? Because this is the one presenting the marker here, and this is the one receiving. So you can have these cells talk to one another, where it is sending a signal to this one, and at the same time, the receiving cell is also sending a signal to the other one. Now, having said that, you find that the most common way in which cells talk to one another is not really through that link up, but through the chemical messengers. So these chemical messengers, we've already encountered some when we were talking of synaptic and neuromuscular transmission. We were talking of the neurotransmitters. However, in some other cases, we may have things that are coming from far place, and the targets or the receptors are located far from the signaling cell. So in that case, we talk of a different chemical, which is a hormone, and we'll look at that again in greater details. Others may not fall into the category of a neurotransmitter or a hormone. They may be chemicals that are being released directly from cells, and the receptors are in the adjacent cells. Then in that case, we'll talk of the chemomolecules and we'll talk of the chemoreceptors. That we discuss things we call chemokines. Oh, I've already mentioned here. So I've talked of the hormones, neurotransmitters, and the chemokines. So I don't have to go through that. Any questions so far? Any question? I hope you are all attentive. Yes?
Yes.
Yes.
Is it Mogisa?
Oh, yeah. I'm not on this. Like some examples of the chemokines. I don't know what you mean there.
Yeah, we'll go through them as we are discussing the different methods here. So I'll go individually into all the methods. So when it comes to the chemokines, I'll tell you. So I will use this diagram to block some of the things and describe what is happening. So let me start off with the gap junctions. I don't think anybody has a problem with gap junctions. So this one is talking about the adjacent. When we have direct intercellular communication, one could be through the gap junction. That one, I don't think anyone has a problem with it. And right now, I've just finished talking about the surface markers, whereby I'll give you a good example. Anyway, I can give even an example for gap junction. Or anybody who can think of an example for gap junction communication? Anybody?
Can I try?
Yes.
The ones we've just studied of the neural communication between the neurons and the muscles.
Between neuron and muscle, there is release of a chemical. So it is not
Oh.
like, yes, Alice?
The use of desmosomes.
Desmosomes, they come into this. Can you see where we have surface markers? Because one cell is releasing some things and then the other one is sensing it and then it's reacting to it. Gap junctions, when we have different cells aligned together and they all work at the same time, because you can see when they are stimulated, all these channels open up, and if there is another cell here, they open up. So instead of acting like a single cell, they act like it is one big cell, and we call that a syncytium. It enables muscles or cells to work together at the same time. So where have you encountered gap junctions where you have such kind of reaction? Yes, Lillian?
In the action of the heart,
The heart.
muscles. Yeah.
The heart, yes, Marie? Patience?
Me too, I was going to talk about the vesicle heart and the vesicle and the extracellular material.
Yes, actually, the heart cell is the best example of gap junction communication together with smooth muscle cells. Even smooth muscles, like when we have the cells of our vessels contracting, they usually contract all at the same time, like it is a syncytium. Why? Because of the gap junction. So think of those two as good examples. When you're thinking of the surface markers, I always give a very good examples of how these surface markers in one cell can influence the surface marker of the other. I will draw for you what we usually have in the lab. We have what we call the petri dishes. You can actually have cells. As long as you give them the right media, you can grow them in the lab in a petri dish. So what you do is you put some cells, you put the media. When you put the media there, let's say this is just a layer of the media. So what I will do is I will inject some cells here at the bottom in the media, and then I will disperse them. So if I look under the microscope, I will see them scattered all over like that. And as long as I give them the correct environment, temperature, medium for growth, I give them the nutrients, they will start to multiply. And the funny thing is, if you look at the rate at which they multiply, they move so fast. So after I keep on changing the media so that I can replace the nutrients, you find it is becoming darker and darker, or they are coming closer to each other because the numbers are increasing. Okay, so what happens is that with time, These cells are going to come closer. As they multiply, they become denser and denser and denser. They start to touch one another. So if you look at the growth rate, initially when there were very few cells, they were growing at a very slow rate. Then they start increasing the rate at which they grow. Now when they become confluent, you start seeing them decline in the number of cells new cells that you see. And soon they will reach a point where they'll stabilize and it will become, the whole dish will become confluent. You'll have no more cell growth, no more multiplications. So what is enabling them to transition to this? So if you look at what I have on this side is time against cell number. So in this case, you are looking at population here. So we have a low population, then it starts to increase drastically, then and it goes and levels off. Why is that? Because when they become confluent, the cells will start to touch one another. Once they start touching one another, if this is the signaling molecule, it will send an inhibitory messenger to this site. This is the receiving molecule. So this will become inhibited, and any factors that will enable the cell to divide will be inhibited, and therefore that cell will stop to grow. Remember, there is also another one next here. So even this one will be inhibited. And remember, even this one will inhibit the growth of that one. So every time the cells come in contact with one another, they stop each other from multiplying. For that reason, you find that the number of cells at confluence is when they are all very well packed and touching one another. When they are at that point, the cells are still alive, but the numbers will not increase or decrease, they level off. Okay, so this is a very good example of how surface markers can influence each other, and they cause a response in the receiving cell. So any question, or I move to the next one? Now I start talking of the intercellular communication. I'll draw two diagrams here so that I cover two different. Let's look at paracrine. Now, when it comes to paracrine, this is where somebody was asking about what are chemokines. So in the paracrine, you find that a cell, the signaling cell, is releasing some chemicals. And that's why it is called the signaling cell. It releases the chemicals into its surrounding. Now, these chemicals that are released into the surrounding, they have two options. They can bind immediately surrounding cells, the neighboring cells. They talk to the neighbors, and they cause a response in the neighbor. That is called paracrine signaling. And a good example is when you have a tissue injury. If you hit yourself, you traumatize some part of your body, what will happen is usually that place swells. You've never asked yourself why it swells. There will be some tissues that will be damaged upon hitting them. Those damaged cells are going to release some chemicals, and those chemicals will cause the adjacent cells also to be stimulated to release chemicals. So all these cells will be stimulated to be releasing the chemicals. One of the resulting events that will happen due to those chemicals, you're going to change the osmolarity in the surrounding media. When you change that, because it might open up some cell channels, it might open up, things will happen. And the end result is that the ECF there will attract a lot of fluid. And this fluid is designed to try and neutralize or to dilute the effect of the damage. So once you start having fluid coming in, it is increasing in that interstitial space, causing fluid swelling. And that's why when you press, you start seeing that that swelling can actually go down, it will sink, but the minute you remove your hand, the water fills back those places and it swells again. Now, second thing is that these chemokines, or rather these chemicals, they also have a tendency to attract some of the immune cells and some of the immune factors from the circulatory system. That's why they are chemicals and they have some kinetic content, and that's why they are being called chemokines. And some of them are like things like the histamine. When you injure, you release a lot of histamine, and one of the things it will go is going to cause inflammation, that swelling of the skin. So the person who was asking for an example is the histamine in case of tissue damage. Now, it is not always that paracrine signaling is occurring due to tissue damage. In some cases, it is physiological. Things like when we have in the digestive system, you're going to see when you're doing digestive physiology, some cells, once they are stimulated by a certain food type, they will cause the release of some chemicals, and those chemicals will stimulate other cells to start releasing the appropriate enzymes to digest that protein. So because we have cells talking to the neighboring cells to release the enzyme, then in that case it is a paracrine signaling. So always think of yourself in your house. If you're making noise and your neighbors are hearing it, then it is paracrine. But then there's also that component whereby you're making noise and you're the one hearing it. So here you are, you release the chemical, but this chemical is in the surrounding and you are the one who is receiving it. So in that case, you will cause a reaction within yourself. Maybe you lower your noise. So the chemokine that you release here may cause events on the same cell that was signaling. Then in that case, it is autoregulation. Autoregulation means the cell is talking to itself. So if it is talking to itself, it is called an autocrine signaling. So the difference between paracrine and autocrine is that autocrine talks to itself, paracrine talks to its neighbors. I've written here because I don't think it is in this diagram, but let me just see what we have here. Yes, it doesn't have autocrine, but this is one method of communication you need to understand. Yes, Anastasia?
In your explanation, do you mean paracrine can be, autocrine too?
No, it will not be. The paracrine signal can also cause an autocrine communication.
Okay, that's what I was asking.
Okay, yes, because the same signal can cause a reaction on neighboring cells, but it can also cause a reaction on itself. Yes, Lillian?
Is it right to say the paracrine secretion takes place in mast cells?
Yes, even those ones, it does happen. Yeah, there are quite a number of cells that you can mention. Yeah. So the next one that I would talk about is the neurotransmitter secretion, whereby one neuron which is far placed it can be far or near. Like when we are talking of interneurons, they are very close to each other. But in some cases, the cell body may be in the spinal cord, but the affected cells are in your toes, the muscles of your toes. So it is the same neuron, but with a long distance. So this could be short or long distance targeting. Remember, all this is in adjacent. The ones we have mentioned, they are adjacent. Then when you start increasing the distance between the cells, then you can have three forms of communication when you put distance into it. It could be the neurotransmission or nervous transmission direct. One neuron talking to either the muscle or another neuron which is far placed. Then in that case, it is neuronal communication. Number two, you can also have a neuron releasing a chemical. But that chemical does not go to the adjacent cell. That chemical is taken up by the blood, and the blood helps to transport it to a far-placed target, and therefore causing a reaction in the target cell, which is very far located from the secreting cell. Then in this case, we talk of a neurohormone. So in the first place, we have neurotransmitter being released. That is a nervous communication. In this other one, you have a instead of neurotransmitter, because it will be taken up by the blood, it is a neurohormone. Anything that is being transmitted, any chemical that is being transmitted in the blood is a hormone. So in this case, where the target cell is far from the secreting cell, and the means of communication is a blood vessel, the secreting cell is not a neuron, then it is, in that case, we are talking of hormonal secretion. Hormones, the secreting cell is far placed from the target cell, and the method of communication is blood, circulatory system. In nervous communication, the secreting cell may be far placed, but its terminals are close to the target cell, and they have a neurotransmitter as the chemical for transmission of the signal. A neurohormone takes into consideration the two components, where the signaling molecule is a neuron, but what it secretes is a hormone. So you end up having a neurohormone. So any question on that? Unfortunately, now I have to leave. So I think I will end at this point, but you'll see that what I have here describes all the events that I have shown you in the diagram. I see 4 hands raised up. Kindly put the questions together, and I will see if I can communicate to you on the chat. I have to leave because I'm in a meeting and my bosses are waiting. I have to go, please. Yes, any urgent question, if you can't put it on the blackboard, sorry, on the chat, for the people with the hands up.
Yes,
Okay.
Madam, I want to ask if you could kindly share the notes on Blackboard.
Yes, I will.
Okay.
So kindly let me leave, otherwise I'm also going to be in trouble. So what I will do, I'll put everything in the recording, and I'll make sure that you get it before the end of the day today. Okay, have a nice one.
You too. Bye.